We're at AIDS 2018. We're here with Sharon Lewin, who's with the Doherty Institute in Melbourne. And you have been our cure updater savant, and we appreciate that. And it's been always a pleasure. There's others who can help to do that, but it's always nice to have you because I think you have a special way of making it in more lay terms, which is helpful to our audience. But tell us about what you brought to the conference this year. Anything?
Yeah, well, we had quite a big delegation from my laboratory and other laboratories at the Doherty Institute, a few young PhD students and postdocs, and some of the work was around shock and kills, an area that we've worked in for a while. What are some new agents that can activate the virus? And one of them actually is an antiviral drug, Maraviroc. We have found not only blocks infection of new cells, it has a other action of being a latency reversing agent. So we think that that's quite interesting because it's clearly a very safe drug to use. It's probably about as good or a little bit better than some of the other latency reversing or shock drugs that we have. And we're now going to look at whether we can combine it with other drugs effectively. We've got we had a really interesting paper on how over the 24-hour day we all have circadian rhythms. Things change, our hormones change in a rhythm. What keeps you awake what happens during the day and what happens at night.
And doesn't work
is
well
all regulated.
With the problems with her having adjusting the time zones and stuff.
No, your circadian rhythm is all knocked out once you start, traveling too much. But we do have all of our bodily functions have a circadian rhythm. And a few years ago we noted that if we collected blood from people living with HIV on antiretroviral therapy in the morning or in the afternoon, the amount of virus we could find inside their cells differed. And so there's actually been a lot of interest in the circadian rhythm and HIV production and T cell trafficking for a long time, but we think this might be related to latency. That's very, very early experimental work where we're looking at how that rhythm is regulated and whether we can exploit it to develop new agents.
This is something that I think we remembered from way back. They always said, always remember to draw your blood at the same time of day every time you do it. Like if you're used to doing it in the morning, just continue to do it in the morning. So
it was a big deal in the early days of HIV where there wasn't effective treatments, and your T cell count was critical of whether you were going to have a good outcome or a bad outcome.
And some people's very, very much did fluctuate.
That's right.
I don't know whether it's daily or...
No, it definitely was related. A whole lot of things influence your T cell count, but it certainly has a circadian rhythm, meaning that it fluctuates. It's can be lower in the morning and higher in the afternoon. It varies over time. Many parts of our immune system have a circadian rhythm. So people have been thinking about that in the context of HIV for quite a while in totally different contexts. So we've become very interested in that because we found in one of our clinical trials, actually of disulfiram as a drug to activate the virus or shock and kill the virus, we took blood from our participants on 3 occasions before they got the disulfiram, and it was to make sure we had a really clear baseline before we introduced this drug. And what we found was that of those 3 time points, the 3rd time point was earlier in the day because people had blood drawn and then took the drug. And that's where we got this whole idea about circadian rhythm, and it's opened up a whole new area of research. The other piece of work that we presented here was whether we could get better kill agents. So at the moment we think of shock, meaning waking up the virus, and kill, the immune system clearing the virus. But there's a few problems with that because we've known for a long time that people's immune system returns to normal after treatment but never quite goes back to normal. We know that the virus can escape the immune system. So what we're very interested in, and there's been a bit of work on this, it's still very early days, not yet, we don't yet know how this will translate in people, is can we use agents that trigger the cell to self-destruct. So if we have a whole lot of pathways in our cells that keep the cell alive or kill it, and can we exploit some of those kill pathways to lead the cell to self-destruct. So that if it sees a HIV virus, the cell will destruct. So we presented some work here on looking at some new kill compounds, and there was another interesting presentation of a kill compound that is looking at a totally different pathway, and that particular compound is now being tested in a phase 2 trial. So this is a new sort of way of thinking about kill, not using the immune system, using the death pathways inside a cell. And then some really more interesting work for your audience, because it's a little bit closer to people's everyday lives, is some a really nice work from a PhD student, Jill Lau, who's being supervised by me and a colleague, James McMahon, trying to understand the community and providers' attitudes to treatment interruption. So it's a really big, important issue in the cure field at the moment, because if we at the moment don't have a predictor for cure, meaning if people stop treatment and their virus stays at undetectable levels for a very long time and then rebounds, like the Mississippi baby, or people stop treatment and their virus stays at low but detectable levels, like the French Visconti cohort.
It's still doing its dirty deeds.
Yeah, we can't predict the outcome. So many clinical trials now have had and will have a treatment interruption phase where people stop treatment and we wait and measure very carefully and closely, and when the virus comes back, we restart treatment. And treatment interruption means different things to people living with HIV who have been told you must take your drugs every single day. Do not.
So now we're telling them not to.
Or we're saying stop it for a while and you'll be okay, and we'll measure it. And and also their doctors as well. If you don't work closely in the field, their doctors are also a bit confused about the message. So Jill did a very large survey of several hundred people living with HIV and people that treat HIV, predominantly in Australia, but she had many participants in the survey from other countries, including the US and Asia. And what the study showed was that the willingness around treatment interruption was far higher amongst patients than their providers. So many people living with HIV were quite prepared. Thought this was a reasonable thing to do in a safe environment that was monitored far higher than what their providers wanted. We also asked them about expectations of cure and how likely we are to see a cure in the next 10 years. And
is that correlated in any way with their energy?
People living with HIV were far more optimistic about seeing a cure in the next 10 years than their providers were. So I think what the work showed us was this difference with what people living with HIV want and what their providers think should be done, and that there's a real need for better education of both people living with HIV and providers around what we're trying to achieve with treatment interruption and what the real risks are. So I think that work was really important. And she also did a very extensive study of all of the publications out there on treatment interruption to get a very good understanding of what the risks are.
We've had a lot of experience with that. Now we're just gone by that. Now we're coming back to it because there's a real practical need. It's maybe essential. At least at this point, we don't see any other way around it.
Yeah, and at the we normally have a scientific workshop before this meeting on cure, but given there's so much going on in cure in the science area, we decided this year that all of that science will be at the main meeting, and we have had lots of really fantastic sessions on cure research here, and a lot of scientists attending. And the workshop we did before the main meeting was a community-based workshop, and we used a whole lot of different sort of formats for that rather than the standard lecture and questions that boring scientists do. And one of the sessions we had Steve Deeks interviewing one of his patients, Clark Housley, who underwent a treatment interruption, and he was a man that started antiretroviral therapy very, very early, probably within about 10 days of becoming infected, was on treatment for a long time. They couldn't find virus anywhere. He was on treatment for about 3 years. And then he stopped his antiretroviral therapy, and Steve and his team monitored him very closely for It was in the end 10 months, and then the virus came back. And this has been reported already in the literature last year, but Clark came out and spoke about his experience of participating in that study, why he did it, and what it felt like, sort of waiting to see whether he was cured or not. And he gave he told a great story, and a few of the main messages was how important it was, the team that was looking after him. He had complete confidence in what they were doing and really wanted to assist, and was very much driven by making a difference to the field.
I think there's a difference there, because you have a highly informed patient and a very informed physician.
Exactly.
So I think,
it was a pretty unique setting,
yeah.
being in San Francisco, true. And then when he actually interrupted treatment, you do have to monitor people closely. Usually what we do in these studies is they may have a viral load test 3 times a week for the first months, and then weekly for the next month, and then it might extend to 2 weekly or monthly. So people have normal lives over that time. So he was I think he went to Disney World for a week or 2, and they organised to get his viral load tested out there, and all the different efforts it highlighted actually that if we're going to do a lot of treatment interruption studies, we need much easier ways to monitor the virus. So like a home-based viral load test doesn't have to tell you the numbers, just has to tell you it's there or it's not.
Exactly.
Is an area that is worth researching and investing in, because that would really improve the practicality of a treatment interruption.
So anything else pop out at you at the conference that bears discussing? Or
Yeah, I think there were 2 really important studies. First was River. River was a study done in the UK of 60 people who received antiretroviral therapy shortly after infection, within 3 months of infection, so during acute infection. And then after several months on antiretroviral therapy, they were randomised to continue their treatment or to undergo shock and kill intervention. And here they used vorinostat, which is a drug that we used years ago, not a fantastic latency reversing agent, but a lot of data on it and safety, and the study started several years ago. So they used vorinostat together with a vaccine to boost the immune system. And then at the end of the study, they measured how much virus was in the blood. So they didn't do a treatment interruption. And the study was it was a bit disappointing, because they saw no difference in the amount of virus at the end of the study, whether you just had antiretroviral therapy or you had antiretroviral therapy with a shock and kill intervention. Quite a lot of discussion about this study. We learnt that the vaccine actually did boost the immune system. They haven't yet really fully finished all their studies to know whether the shock drug shocked the virus. And because they haven't stopped treatment, we don't really know whether it had an effect, because when we measure the total number of infected cells with HIV DNA, it's not a very accurate measure of how much virus is in someone's body that we need to worry about.
So if you don't have the shock, which gets all of the virus out of the compartments, you're not going to be able to kill it in a way that you hope to kill it.
That's right. There's a few reasons why, when you put 2 things together, why the study may not have the outcome you hoped.
So
the shock drug may not have been strong enough. The kill drug may not have been strong enough.
So the compounds may have been okay. It's just that you didn't have the formula down right.
Yeah, we don't yet know which was the bit that worked or didn't work, and why didn't they work effectively together. So it's a really important study. It is the biggest study of shock and kill. It's randomised, meaning that you had one group of people that just had treatment alone and antiretroviral therapy alone, and the other that had antiretroviral therapy with the shock and kill drug. It's not at all. I don't think, and certainly the lead investigators, Sarah Fiedler from the UK, and many others don't see this as the end of a shock and kill strategy at all. A really important study, highlights how difficult these studies are, how long they can take, because you have to know each of the agents are safe on their own. And I'm hoping with more analysis, we'll learn quite a bit from that study. The other important study was also negative, but a very important study, and that was using an antibody called vedolizumab. This is an antibody that's used for people with inflammatory bowel disease, and it alpha 4 beta 7 it's also called, and it stops T cells from moving into the gut, and we know there's a lot of virus that persists in the gut.
Gotcha.
And there's a lot of excitement 2 years ago when in a monkey model, monkeys with infected monkeys with a relative of HIV, something called SIV, were on antiretroviral therapy. They received this antibody, and then antiretroviral therapy was stopped in the monkeys. And it was a very high-profile publication because the drugs that received the anti-alpha 4 beta 7 or vedolizumab controlled the virus and the monkeys. They repeated that study, and in a monkey study, exactly the same virus, exactly the same antibody, and no effect. So we're still trying to understand that. And then Tony Fauci presented the clinical trial of people living with HIV on antiretroviral therapy who received the antibody before and during treatment interruption. And again, the virus rebounded, and it looked like they really had no effect. So we're all trying to understand that data and why the two monkey studies look so different.
You don't want to throw the baby out with the bathwater. If there's a chance you can make it work by just tweaking it a little, it might.
And after a very small monkey study, many investigators, moved on to human clinical trials. They'll be informative. And we know this is not an unsafe intervention. The drug has been around, been given to many, many people, but we want to be doing, clinical trials that we think we're going to have an outcome. So I think the repeat monkey study was very important. There's another monkey study happening with a different virus. We're seeing a lot of exciting work coming from monkey studies at the moment. We don't yet know if they will translate into human studies. And in the monkey models, it depends what monkey you're using, what the genetic background of that monkey is, and really important what virus you're using, because there are a whole different viruses you can choose from, and some viruses might be more easier to control than others. So there is another monkey study using a different virus, which I think will be very informative. And some of the clinical trials, I believe there's 3 clinical trials currently still underway, and we'll have the results. And really, when all that's together, we'll know whether this is a path worth pursuing.
The problem, as you stated earlier, is that there are many steps in developing even one trial. So it's good that we have them going on concurrently, because we're hoping that somewhere there's going to be some wins or some knowledge that can. And the other thing is, a lot of this information, obviously you know it and it's being presented, but proprietary does exist. But it's good to get this out, and it's good that we have this collegial relationship in the field of cure.
Yeah, negative studies are very important for us.
Very helpful.
And actually at the end of the session where both of those studies were presented, one of the funding agencies here from the Netherlands, the AIDS Fonds, one of the senior members of their agency, stood up saying how important it is to present these negative studies, of course, because none of us want to be working on things that we think aren't going to work.
But the population needs to not be depressed by that, because that is a learning piece.
Yeah, that's right, and it shows where our efforts should go. I think with River, there's the investigators, worked really hard to get that presented here. They've looked at their primary endpoint, but whenever we do a study, especially an early investigational study, there's a lot more. We have one endpoint that we designed the study on, but there's loads of information still to be gleaned.
Out in the end,
And that's happening now.
Well, thank you so much, Sharon. I really appreciate your time, and it was a great talk because it covered a lot of territory. Thank you so much.
Pleasure. Thanks a lot.
Yeah.